Sorry.
I think you're going to have to get them because it's not showing up. Okay, let's get started. Having trouble with the laptop showing up, but we can do without that for a few minutes. I put this slide up because it's important, obviously, for you to know that Tuesday's lecture was the end of Quiz 2 material. That's going to be next Tuesday. It's the same setup as we did before. One way you can save time is don't read the instructions. You will not hear another professor tell you to not read the instructions, but you don't need to because all the practice quizzes I've given you, it's the same instruction set. So there's really no need to waste time reading those instructions. If there's any differences at all, I'll let you know. Go straight to the test material because some of you had trouble finishing, and so that's something you don't want to waste time on. Okay. It's going to be the same setup, though, about the same number of questions, but it will be comprehensive. It will include Quiz 1. Quiz 2 material as well, but the focus is on the new material, obviously. So, any questions about how the test is set up? I'm going to send a reminder of stuff that's due this week, but really what you should be focusing on now is not the group stuff, is the material for Quiz 2. So I would remind you what is due for next week. Or I think Friday we have a couple of deadlines, so we'll go from there. All right, so we're starting a new topic, and it's Chapter 12, which is intracellular compartments and protein sorting. This is sort of the heart of cell biology because we've talked about how you get information decoded from the genome and it goes through the processing, goes to messenger RNA, gets translated into protein. In most cell types, you don't locate the function of a protein by moving the messenger RNA. That's a pretty odd case that happens during development where the protein is localized where the messenger RNA goes. Usually you make the protein and then you tell it where to go, and that determines how that protein functions. So this is a really complicated story, and we're going to spend about 4 lectures on it, okay? And so why is it complicated? Well, we need to be able to, by looking at the protein, where it functions, be able to tell where did its synthesis start? All proteins, of course, start on free ribosomes, but where does it end? If it ends on a free ribosome, generally that protein will function in the cytosol. So in the absence of targeting information, rule number 1 is you're a cytosolic protein, okay? If you're not, then you need to be told where to go. And so we need to understand what those signals are, and most of those signals are actually part of the protein sequence, okay? So it's got a zip code somewhere. Why is this a complex issue? Well, you know, this is a typical cell. It doesn't really look too complicated. You know, you have the nucleus, ER, Golgi, cytosol, lysosome, and organelles. But if all the proteins are made in the cytosol, which is true, all of them are made in the cytosol. Now they don't end up being in the cytosol, but they all start out there. Okay, how do you get it to go from here to all the correct locations? Because if it's in the wrong location, the function is misguided, so it has no function. Let me get this started so you can take a look at the notes. All right, that's going to come up in just a second or two. So this is a cartoon, right? It's simplified to help you understand it. This is what it really looks like. So this is a transmission electron microscope. It's pretty much standing room only. Things are squished together. So this is a complex problem of getting protein to start out here somewhere in this little space and then go to the correct compartment. In fact, there's a pretty good video that on your DVD that I'll have you take a look at. Let me bring this up real quick for you. This one actually works, so you don't have to go to the web. Come on. We're going slow today. All right, so that's where we are. Chapter 12. And so let's take a look at that video right now. And pull that down. All right, let's go back to the menu and try it again. I tried it this morning, so it should work. But you never know. That's awfully fast. Okay, let's forget about that one. And it's probably one of those that actually is on the web, so we'll go back to it. But anyway, if you look at just the little what was coming up here, you can see that it's actually a three-dimensional diagram of all the different organelles, and they're colored for you to see where they are in 3 dimensions. The upshot of this whole thing is it's a complicated request to tell a protein where to go correctly. Okay, but somehow it manages, and so let's try to figure out how that problem is actually solved. And so that's really the basis of today's lecture. So now this is a real cartoon. This is going to be the system that we're going to focus on first, which is the so-called endomembrane system, and it consists of a few discrete components. The ER, which you know is the nuclear envelope as well as the ER. The Golgi, all the transport vesicles that are involved in moving proteins around inside of vesicles, the endosome, and then the lysosome are all components of this internal membrane system, okay, that delivers proteins to particular organelles, all right? Now, so there's three different mechanisms of getting proteins to where they need to go. The first is called gated transport. This is sort of like a British subway map of the UK subway system, and some of your textbook authors are actually British. That's why they use this system. But the gated transport actually occurs in a very different way for proteins than it does for ions. Okay, so we're not talking about ion gates. Ion gates are really tiny compared to the gateway between the cytosol and the nucleus. So that's where gated transport is going to occur. And the gate that they're talking about is through the nuclear pore. It's a very special type of gate. Right? So that's what we mean by gated transport. Now the next place to go is from the cytosol, because everything starts in the cytosol, no matter what. All synthesis happens in the cytosol. And it goes to membrane compartments. Okay? How do you get a huge protein across a biological membrane? You cannot do it using a transport protein. It's not going to work. So what you need to do is to be able to open up a pore, basically, and thread the protein through the membrane. Now, that type of so-called transmembrane transport occurs when you send proteins to the ER, to other organelles like peroxisome, plastids in plants, mitochondria. Our poster child for that process, which is very similar between these different compartments, is going to be the ER. We're going to focus on how you get proteins across the membrane into the ER. And with small changes, you can study that in Cell 2. Those of you that take it, they will go beyond this and talk about how you get things into mitochondria, chloroplasts, plastids, and other organelles. But our focus is going to be transmembrane transport into the ER. Okay? And the third type of transport we'll look at is vesicular transport. And that's a very special type of cargo that you're going to take cargoes and you're going to load it up and butt it off into a vesicle. So here's the donor compartment. This could be the ER. And so you have some cargoes that, like insulin, for example, that you need to secrete outside the cell into the blood, and you package it into a vesicle. And so we have to understand what causes vesicles to protrude off that donor compartment and form this vesicle. That vesicle then displays targeting infusion proteins to say, this is where I'm going to go, and this is how I'm going to fuse. All right? So it goes to a target compartment, and then these special targeting infusion proteins allow it to fuse with the correct compartment. That's a pretty amazing process. Okay? So then it fuses with the compartment, and then the vesicle delivers its contents into the new compartment. That's called vesicle fusion. So whatever started here is going to be transferred. It never crosses a membrane. It's membrane fusion that causes the two compartments to come together. Okay, and we'll get to that as the last topic in this series. All right, so the first thing we want to look at are some general considerations. I've mentioned one rule, and that is all proteins start, they all begin on ribosomes that are free, and it all begins in the cytosol. Okay? If you're going to these particular places, and we really need to modify this location, nucleus, because nucleus could include the nuclear envelope as well as the nucleoplasm. So really what you should do is mark this out, and I'm going to correct it and then post the change. This should be the nucleoplasm. If you're going to go from a cytosol into the nucleoplasm, you're going to use gated transport. But your synthesis starts and finishes on a free ribosome. That ribosome never touches the ER. Okay? All right? And that's true for anything that's just going to be chunked into the cytosol. So if you're a ribosome and you're making a protein that's destined for the cytosol, it has no targeting information because the default pathway is the cytosol. So there's no instructions about where to go because you're already there, right? So you don't need any targeting information. Now, if you're going to go to the mitochondria or chloroplast, this is a little crazy because what happens is the protein begins on a free ribosome and ends on a free ribosome and is dumped initially into the cytosol, but then there are targeting sequences that take that protein by a very complicated process and deliver it to those compartments. So we're not going to cover the mechanism, but it's important for you to know that those proteins that go to mitochondria, peroxisome, chloroplasts, are those organelles. The protein starts on a free ribosome and ends on a free ribosome and is dumped into the cytosol and then targeted to those compartments. Okay, usually the generalization you would make broadly is if it's going to be a membrane protein, it's going to be targeted to the ER. So it would start on a free ribosome and then it would finish on a ribosome bound to the ER. But that's not the case for all membrane proteins. If it's a peripheral membrane protein that is on the cytoplasmic side of a membrane, it's going to start on a free ribosome and finish on a free ribosome. Okay? So what's a good example of a membrane protein that's peripheral and operates on the cytosolic side of the membrane? Let's think about the plasma membrane. Can you think of any poster children that are going to be on quiz 2 that would fit that definition? Peripheral, not integral, and it's on the cytoplasmic side. There's actually a bunch. Yeah? Annexin. Excellent. So that is it's peripheral. What brand of peripheral protein is it? What's the two categories? There's two. Direct or indirect? Which is it? Is it direct or indirect? Direct. And that's because it binds to the phosphatidylserine hit groups. Right? So that's a direct. So that protein, even though it associates with the membrane, does not go through the endomembrane system. It starts on a free ribosome. It's finished on a free ribosome. It's just dropped into the cytoplasm, and there's information that tells it to go to the plasma. Okay. So this is not always simple. Membrane proteins all go through the endomembrane system. That's not true. All right, so here's the next category is this. If you're a protein that you're targeted for secretion, plasma membrane, or the following locations, you're going to begin your synthesis on a free ribosome, and then that free ribosome attaches itself to the ER. That's what we call ER-bound, ER-attached, or the rough endoplasmic reticulum. That's why the endoplasmic reticulum looks rough, is because ribosomes are attached to it. And they're making proteins destined for these compartments. The ER, the Golgi, the lysosome, secretory vesicles, plasma membrane proteins, integral or exoplasmic peripheral membrane proteins. So this is a little confusing because some peripheral proteins operate on the exoplasmic compartment, correct? Do we know of proteins that would fit that definition that are going to be on quiz 2? Yes, we do. Think of an example. It's got to be a peripheral protein. Pardon me? Anchorin's on the cytoplasm. So that would have fit that definition. So that's a good one to remember. All the actin cytoskeleton, all those indirect peripheral membrane proteins that are on the cytoplasmic side, they go in this category there. But what goes here? Exoplasmic peripheral protein. Pardon me? Glycolipids are in the plasma membrane, okay? And so they actually get there through vesicle trafficking. So, but we're talking about proteins.
Are the calcium binding proteins on the exoplasmic side? Calcium binding proteins?
Calcium binding proteins, basically on the extracellular side are proteins that bind calcium are like albumin and things. They're non-specific. Most of the specific calcium binding proteins are cytoplasmic. What about, okay.
What about glycoprotein?
Well, you would have to say sort of extracellular matrix protein. All of them out there that bind to the cell. Because remember, that's how certain proteins that are integral get frozen in place because they attach to the extracellular matrix. So all of those extracellular matrix proteins are going to be in this category because they're secreted. Now, what about a GPI anchored That's a lipid anchor.
Like alkaline phosphatase?
Yes, that's a good example. Anything that has a GPI anchor is going to be made by this system because it has to get into the exoplasmic compartment. All right. Now, are there lipid anchored proteins that would fit in here? You really need to know these before Tuesday. Not because of this, but because of quiz 2. RAS and SARF. Okay. So those would be made on a free ribosome, and then they're finished on. They start on a free ribosome, they finish on a free ribosome. They are not shunted into the endomembrane system. So it's kind of clear, but it can be sort of dicey for particular proteins. All right. So we talked about this roadmap. And I mentioned the fact that if you get targeted to one of these areas up here, you're going to have a piece. You're going to have a zip code. If your zip code is not there, where do you go? You should say this really fast. Cytoplasm. Okay, just no information. You're going to go in the cytoplasm. You have to be diverted from the default pathway not to go in the cytoplasm. What diverts you are these targeting sequences. Okay, so nuclear proteins have, surprise, a nuclear localization sequence. Sometimes it's called, the S stands for nuclear localization sequence or signal. Those two terms, signal and sequence, are interchangeable, with one exception that I'll point out. But NLS, nuclear localization sequence, is a tag that says that protein is going to go into the nucleoplasm and function. So what poster child do we have that must function in the nucleus? What's in the nucleus? Pick an example of something that's in a nucleus. DNA. What binds to DNA? I'm sorry? Pole 2, yes, right. So big proteins that do function, that function in the nucleoplasm would have to be targeted there. What about transcription factors? They're all made in the cytoplasm. And actually, one way of keeping them inactive is to keep them in the cytoplasm by hiding the nuclear localization sequence. We're going to see an example of that today. Okay? So if you reveal the nuclear localization sequence, that gives it a permit to go in. Now, what if you have an opposite sequence? You can have a protein that gets in the nucleus, but it has to get out. So that targeting information would be a nuclear export sequence, or an NES. It's a different sequence. Okay? So, and we'll look at some specifics, but so here's the basics for this. Secreted proteins, they're going to go into the ER, the Golgi, that whole pathway. The endomembrane protein have what's called a signal sequence. And this is a very special term that only applies to proteins going into the endomembrane system. That's because this was the first one that was discovered. So the signal sequence is on the N-terminus of a protein that's going into the ER. And then it may go further in the pathway depending on what other information is attached to the protein. Okay? So that signal sequence is very specific. It's an N-terminal sequence. Generally it has, this part is pretty fuzzy here. It might have a couple of basic amino acids, but those basic amino acids are going to be adjacent to a central hydrophobic patch of about 6 to 12 amino acids. That's called the signal sequence. Okay? And then there's a couple of polar amino. So item 1 and item 3 are a little bit fuzzy. But this one, this central patch of 6 to 12 hydrophobic amino acids at the N-terminus of the protein is very specific for a signal sequence. Okay? And a signal sequence allows you to get into the ER, and then you can do your work there, or you could go to the Golgi, or you could go to an endosome, or you could go to the lysosome, or you could go outside the cell and be secreted. So a good example, a way of thinking, a protein that goes through all of those compartments is hormones. Okay, so the hormone insulin or growth hormone is got a signal sequence. It tells the ribosome to start making it, and then it exposes the signal sequence, and then that is actually read by a special set of proteins that then thread it into the ER. Once it gets into the ER, it then buds into a vesicle that goes to the Golgi. and then it goes through all the Golgi stacks, and then it goes to a special compartment, the regulated secretory vesicles, and awaits a signal to be released. If you're a secretory vesicle and you've got insulin, what's the signal you're waiting for? You're in the pancreatic beta cell. You don't want to, do you secrete insulin all the time? Full blast? When do you secrete it? A little physiology here. Yeah, after a meal. So when your blood glucose goes up, the beta cell is waiting, and it detects rising blood glucose, and then it tells that secretory vesicle to fuse with the plasma membrane. So that's why we call it the regulated secretory pathway. So we'll get to how it gets to that position. But it all started with getting you into the ER, and to do that you had to have a signal sequence. Okay? We'll call nothing else by those two numbers. Signal sequence is always at the N-terminus to get you in the ER. Okay? Now, understand that mitochondria, chloroplasts, and peroxisomal membrane proteins possess their own unique targeting or transit sequences. We're not going to talk about those. But all of those proteins that go to these compartments are first made in the cytosol in toto. They finish their synthesis in the cytosol, and then the targeting sequence tells it where to go. That's very different than going to the ER and then going through this series of membranes. What would be an example of a protein that you know about in any of these organelles that must go to those compartments? You must know of some. I'm sorry? Exactly. Okay, so the one we already know about is the FOF1 ATPase, that's ATP synthase. Where does that function? Inner mitochondrial membrane, right? To get into that compartment, what's happened is you make the protein and then you send it to the ER. Okay. All right, so let's go on with the generalizations. So in addition to, once you get the protein into the compartment, there may be additional what are called topogenic sequences that determine its topology in the membrane. What does that mean? It means that particular membrane proteins face a very specific place. For example, quiz 2 material, the GLUT family, the N-terminus and the C-terminus are facing the cytosol, right? That topology is determined by topogenic sequences that tell it to be oriented that way. If it was oriented the opposite way, it would not function. So there's we'll get to that whole topic, but understand, once you get there, that's not the end of the game. You have to tell the protein how to fit into the membrane. Okay, so you can see this gets complicated rather quickly. So let's start with an easy topic, and we'll, this is just a table that shows you some examples of these sequences. And so if you want to get it imported into the nucleus, that's a nuclear localization sequence. You've usually got a couple of prolines, and then you have a patch of positively charged amino acids. Okay? And if you want to get exported from the nucleus, it's called a nuclear export signal. And it has alternating Lu something, Lu something, Lu something, Lu something. So it sort of skips a little thing and has lots of leucines in it. Okay? The one that we're going to really focus on the most is import into the ER. That's called the signal sequence. And so it's got this hydrophobic patch. That's its strongest characteristic. It's got some polar or charged amino acids on either side. Not always. In this case, there's no positive amino acids, so that breaks that rule. So the most defining rule is this patch of 6 to 12 amino acids that's close to the N-terminus. That defines the signal sequence. And this is really the only one that we're going to focus on until we get further into the endomembrane system. Then we'll learn this one. Okay. So there's targeting information that's in the protein itself. And our first example is going to be gated transport. We'll start with the simplest. You won't think it's simple, but it is. It's the simplest system. All right? So we're going to go from the cytosol. And if we want to go into the nucleoplasm, not to the nuclear membrane, we want to go into the nucleoplasm. Let's say we're talking about transcription factors. We're going to have to use gated transport to get into the nucleus. And that type of transport is mediated. It's got to get across the nuclear envelope. We've got to get past, not into the nuclear envelope. And you know what the nuclear envelope is. We've talked about it already. It's just the ER that's squished on top of the nuclear lamina, right? So this is the nuclear envelope. And so you have a space there, the perinuclear space. But you've got all these nice little holes in here. And these holes are not just little punched out things. There's a structure there that's called a nuclear pore complex, NPCs. Okay, so that's where the protein is going to go. The ER membrane, as you remember, is attached to the nuclear lamina. That's the intermediate filament grid to which chromatin is attached. So chromatin is attached to the nuclear lamina, which is attached to the nuclear envelope. So we've got to penetrate this whole thing. And what allows us to do that is this ugly-looking thing. I sort of call this the hairy drain model of nuclear pore complexes. And so this nuclear pore is a huge group of 30 different proteins called nucleoporins. And fortunately for you, we're not going to memorize all of them. Okay, so nuclear porins, subunits, make up this structure that has 8-fold symmetry. It's got fibrils that project into the cytoplasmic side, so if you look at a nuclear pore complex, you can always tell where you are. The cytoplasmic side has fibrils, and those fibrils are actually bound to very important proteins. Okay? And the inside has what's called a nuclear basket. We're not going to worry about its structure. It's just there. It's projecting into the nucleoplasm. Okay? I said it has 8-fold symmetry, and if you look at an EM, you can really see it. That's not made up. They actually determined the structure quite precisely. So this thing has actually got a bunch of proteins, and things go in and out. Okay? So how do things go through this large structure? Okay? In fact, you can see, so we know that this is the nuclear side because this is the nuclear basket projecting into the nuclear plasm. And then that side is the cytosol. And you can't see those fibrils because they're real small and real thin. But this thing is a really large looking thing. So trafficking is going here, not through here. And what would this thing be that's stuck here? Ribosomes. Because this is the ER, right? It's two things. It's the ER and it's the nuclear envelope. Okay. That's a side view. So here's the rule. If you're small, you're less than 9 nanometers in diameter, you get a free pass into the nucleoplasm. So glucose and amino acids and salts, calcium, sodium just diffuse through. It's simple diffusion. Okay? If you're small, you simply diffuse. Right? What happens though, if you get up above 9 nanometers, then you have to be actively transported by a mechanism that gets you across this thing. Actually, they split ATP, and you move proteins that are as large as 39 nanometers in diameter. That's the cutoff. Anything bigger does not go through. So if you're bigger than 9 nanometers, or you're small, or you're equal to 39 nanometer, about 40, you can be transported, but you have to use active mechanism to transport. It differs from anything that we talked about. It's not like there's no pump. Okay. What actually happens is the protein that's this big to go through this thing has to be bound to a receptor, and then it's trafficked through that nuclear pore. Okay. Let's look at an experiment to see how these targeting sequences change the location of a protein. So we have a protein that's called the T antigen. This is the T cell antigen. And that T cell antigen operates in the nucleus as a transcription factor. Okay? And as long as it's got this nuclear localization sequence, and we can attach a fluorescent probe to this to track its movements, what happens is it goes into the nucleus. You can't even see the cytoplasm. So all the fluorescence, after a period of time, ends up in the nucleus because this thing has been decoded by what's called an import receptor, and it's been taken through the pore actively and placed into the nucleus. Now, what if you go through and you mutate one of these sequences, you mutate the DNA, and then you tell the cell to make it, okay? So now it's got a single threonine replacement of one of these lysines. It's no longer qualified to be a nuclear localization sequence. You've destroyed the targeting information. And what happens is, as soon as the protein is made in the cytosol, it has no way to get out of the cytosol, and the nucleus remains dark because it won't go in there. So that's how important the nuclear localization sequence is. You cannot fool with it. If you hide it by having another protein attached, this will also happen. It will stay in the cytosol until you unmask that sequence. So that's a control point in controlling transcription factors. Yes?
So if you do mutate it like that, it stays in the cytosol, does it get degraded? Or
After a time, it would get degraded, but sometimes they hang around for certain, they have a half-life.
okay.
And if they're not used, they will be degraded. And that degradation usually occurs in the protein cell. All right, so where does that bring? It brings us to the mechanism of getting this in through the pore. Okay. So I mentioned that the nuclear localization sequence is read by the nuclear import receptor. Okay? And what that is a series of proteins that recognizes this is the cargo that's going in. This would be like the T antigen or any transcription factor would have this part right here. Now, is it exactly the same? No, it's not. This is shown in cartoon fashion. It could be this, slightly different shape, but they're all recognized by this little S-shaped, and that's a cartoon too, an S-shaped looking nuclear import receptor that detect. What's its job? Is to read the nuclear localization sequence. It's a code reader, basically. But in this case, the code is the targeting sequence. Okay? And so, if you have this as your license to go in, you're allowed to be transported into the nucleus, okay? And sometimes there's another adapter protein, but so we're not going to dwell on that. It gets more complicated depending on different proteins. So our example, we're going to focus on this simple kind of thing. You have a protein import receptor that recognizes the NLS, and then this is going to happen. Okay? So active transport through the nuclear pore is driven in a certain direction by an old friend of ours, the GTPase molecular switch. So let's remind ourselves of how this thing works. Okay? If you have a protein that's got GDP bound to it, this is called, and the protein that's the switch is called RAN. And that's a really good name because it runs it in and out of the nucleus. Okay? So this is another protein that's involved in transporting the cargo and the import receptor to do its job. Okay, so RAN GDP is clearly in the off state, right? It goes into the nucleus by a process that we're not going to worry about. Okay, and once it gets there, what it does is it runs into the RAN GEF. All G proteins, GTPase switches must bump into a GEF. The GEF is the guanine nucleotide exchange factor. What's an example that's going to be on your quiz too? Sorry? The eukaryotic initiation factor 2B. Okay. Right? It's the GEF for the eukaryotic initiation factor 2. Right? So this thing has to bump into a GEF to do what? That kicks out the GDP, it picks up GTP, and that turns it on. Okay. Now, what makes this directional transport is the GEF is located attached to the chromatin. And so this is always going to be in the nucleus, correct? And then in order to get back out and to be turned off, it has to bump into the RAN gap, GTPase activating protein. That's attached to one of those little ugly fibrils projecting into the cytoplasm. So because you put the RAN gap and the RAN GEF on opposite sides of the membrane, you've now dictated direction of traffic. So how does that work then? So here's the rule. Okay. Well, this is just reminding us that this is just a GTPase switch that you already know about, right? And it's just like a phosphorylation switch, except you need the GTP means it's on, GDP means it's off, okay? All right, so let's focus on this system right here. So we're going to start in the cytosol with a, let's say, a transcription factor that has an NLS. What recognizes the NLS? The nuclear import receptor, shaped like an S, okay? And so it binds to the cargo, and that's all it needs to get into the nucleus, okay? How does it do that? The nuclear import receptor is actually binding to the nuclear porins on a motif called an FG repeat. Phenylalanine glycine repeat is a sequence that is a little hand grips that it actually travels through the nuclear pore. Okay? What's reading that? It's actually, the cargo can't read it, but the nuclear import receptor can. It's reading FG repeats. And so it's like a series of monkey bars swinging through this pore. So that's how you get big things that are less than 39 nanometers in diameter, but they're greater than 9 nanometers. So it's too big to diffuse, but it's small enough to get through the active transport mechanism. So it swings through this, and it gets into the nucleoplasm. The problem is you can't let go of the cargo until what happens? Until you bump into RAN GDP. It's the key that's going to unlock the cargo from the nuclear import receptor. So the RAN GTP binds to the nuclear import receptor, and that causes it to release the cargo, and voila, you've transported the cargo from the cytosol into the nucleoplasm. And then it can do its function, which is transcription factor, binds to DNA, using all those nice motifs that we talked about. Okay. So the role of RAN GTP in the on state is to release imported cargoes into the nucleoplasm. Okay? And then its second role is it's going to take the only way you can, your only license to get into the cytosol, that's rule number 2, is RAN GTP must be attached to you. Okay? So the nuclear import receptor is taken out back to the cytosol because it's bound to RAN GTP. Now, how do you get RAN GTP to release the cargo, which is the nuclear import receptor, back to the cytoplasm? You bump into the RAN gap. Okay? The RAN gap is attached to what? That fibril that's always on the cytoplasmic side. So what that does is it tells RAN GTP to split GTP, release the phosphate, and return to the off state, which is RAN GDP. When it does that, it changes its conformation, and the switch lets go of the nuclear import receptor, which is free to take another round of cargo transport. So that completes the whole circle. The only thing that's missing here, the book is not going to tell us, which we'll cover later in cell 2, is how does RAN GDP get back into the nucleus? We're not going to worry about that. It just does by separate things. Yes?
So the system which that, like, first
well, it involves a conformational change. It's all about the molecular switch causing the conformational change. First of all, it tells, and it's nice that the cartoon is sort of S-shaped because it's got a binding site for this cargo, but that other binding site is for RAN GTP. And so that switch has to bind to it to tell it, first of all, let go of your cargo. And so it delivers the cargo. And then now it's going to go back out. Its license to leave is because it's bound to RAN GDP. That's its license to leave. Okay? And so it gets back out, and then RAN is not going to let go of it unless you split GTP back to the GDP state. So it's a different kind of switch. Okay, let's take a break there. Yeah.
Where does the RAN gap protein function to rephosphorylate?
Well, it's going to take. Well, see, you can't rephosphorylate it because it actually never rephosphorylates. It's actually an exchange reaction. So distinguish between them. One is phosphorylation. This is just like getting rid of GDP. So it's a association reaction, and that occurs when it goes. It goes through by a mechanism in a hand wave. Okay? So it comes back in here in this GDP state, and then it bumps into the chromatin that's holding on to the brand gap, and then it's going to dump the GDP and pick up GTP, and then it's ready for another round. Yeah.
Is that okay?
Yes, it's fine. I can clean up. You don't have to come back. Yeah.
I need this from you.
Oh, okay, sure. Good.
Topic, did you receive it or you haven't checked it?
It's probably because I had to give a departmental seminar yesterday, so I was a little bit busy with it.
But we decided to go on.
And you send it to Blackboard.
Yes.
So that's all you need to do.
That's all we need to do, and then it's going to show up for our group.
It should. The key thing is when you send email that's representing a group, you should put the group name in the subject line.
Okay, yes.
But if you didn't, it's fine. But it makes my job easier, so I go, okay, here's group 12, and I can just look at the names of the files instead of opening them and hunting for what group you're in.
Okay, well,
Yeah.
they're in the topics. I mean, I think I did. I don't remember.
It's okay. So that's a good. I may try to remember that now that you reminded me. Is when you send those, try to put your group number in the subject line.
All right, thank you.
Good.
Called the VO pump we talked about.
Yeah, VOV1.
Right. Mm-hmm. So does VO have two functions to pump out protons and it can also bring in protons? Because I know we have a cell on the inside. There's a vesicle, which also has a VO as well.
Right. But it's always doing the same thing, and here's the rule: no matter whether it's in a vesicle or in the plasma membrane, it's pumping protons from the cytoplasmic compartment to the exoplasmic compartment. Because the inside of a vesicle is the outside of the cell. You should think of that exoplasmic compartment, no matter where you see it, is the outside of the cell.
Okay.
So it still has the same orientation. And what's our rule? The ATP binding domain must face the cytoplasm, because that's where it's picking up ATP.
Right.
But the direction of pumping is always the same, cytosol to exoplasm.
Okay.
And if you remember that rule, then you won't get confused, no matter what compartment it's in, because it's in the ER, it's in vesicles, it's in the lysosome, it's in endosomes, and it's on the plasma membrane.
All right, that makes sense.
Good.
All right, thank you.
Okay, why don't we get started? Okay, so we've talked about one half of the equation, and that is importing cargoes. What if you need to be exported? Okay, and we'll see an example of a protein here in a few moments, that if you have to go both directions, you have to have both an NLS and an NES, a nuclear export signal. But let's say, for argument's sake, that we have a cargo in the nucleoplasm that finds itself there that needs to be re-exported back to the cytosol. So it has to have a nuclear export signal, and it has leucine and some amino acid with leucine. So it's got a bunch of leucines that skip residue. Okay? And what happens is that allows it to bind to a nuclear export protein. And it's also shaped like an S, just for cartoon purposes. But what does it do? Well, what it does is it binds to RAN GDP, the nuclear export receptor, and then that allows it to pick up the cargo. So it must bind to RAN GDP to be exported. That's what the export receptor does, and then it picks up the cargo. All right. So now it's actually got. This is a hetero trimer, right? It's got the nuclear. It's got the export receptor, nuclear export receptor, which is the S. It's got the molecular switch, which is RAN GDP in the on state, and then it's got the cargo. Okay, that has the nuclear NES signal. All right, so then that's allowed to climb through the nucleoporin monkey bars, and the license is you cannot leave the nucleus unless you're attached to RAN GDP. That's true for this, and it's true for cargoes that are being exported. So that's a basic rule. To get out of the nucleus, you must be associated in some way with RAN GDP. So it leaves the nucleus, and then it bumps into these little fibrils, which is shown here. The fibrils have bound to it RAN gap. Okay? And RAN gap, as you remember, if we just flip back to the switch system, is this part right here. So if it goes here and it does its job, and then it leaves the nucleus, and if it bumps into a RAN gap, which is attached to the fibril, that will split the phosphate off and return it to the what? GDP off state. And when it does, it releases the nuclear export signal, right? Nuclear export receptor. And so it releases that, and the nuclear export receptor releases the cargo. So again, RAN is involved in two things: trafficking things through the nuclear pore and releasing cargoes from the receptor. So it's got a couple of different jobs. But what makes this travel in the correct direction is the state of the switch and the placement of the gap out here and the GEF in the chromatin. So that's also a key. Where you put the gap, where you put the GEF. Okay? Questions about how that works?
Did you say it attached the phosphate to the...
No, it takes the phosphate off of GTP, and so it's actually a dephosphorylation reaction to convert it back to the off state. But don't make the mistake of thinking the on state is created by a phosphorylation reaction. That's not true. It's an exchange reaction where it loses GDP and picks up a new molecule of GTP. There's never a phosphorylation reaction. So that's an easy mistake to fall into. Okay, now let's use this to do something medically important. Okay, so here we have a protein. This is a transcription factor, like the ones that we talked about. This is nuclear factor of activated T cells. And what do T cells do? T lymphocytes. They are part of the immune system, right? Do we have any burgeoning immunologists? Yes.
They help macrophages and B cells and also the larvae cells.
And also they're involved in rejection of self and non-self, right? So they attack non-self things and destroy them. So it's an attack and destroy mission. Okay, but that you don't want that happening if it's your tissues, right? So this is turned off under most conditions. How do you turn it on? Well, some signal tells the cytoplasm to release calcium. So you get this burst of calcium, and that activates the T cells. So this is nuclear factor of activated T cells only. So we're going to activate the T cell by developing a burst of calcium, and we'll talk about how that happens later. But there's a calcium signal, and what that does is it turns on a protein that ultimately activates this protein, calcineurin, which is a cytoplasmic protein. Okay? And what calcineurin is really, its activity, it's an enzyme, and it is a protein, not a kinase, it's a protein phosphatase. And its job is to do two things. Number one, it cuts off these phosphates that are masking the nuclear localization sequence. That is what's keeping this T cell from chewing up your tissues incorrectly, right? It's turned off because its nuclear localization sequence is masked. It's hidden by the phosphate groups. So if you want to reveal them to the world, what you do is this protein phosphatase is going to come and clip them off. That reveals this little nuclear localization sequence, okay? And simultaneously, it's going to mask the nuclear export sequence. So it's going to remain in the nucleus once it gets there until it's told to leave, okay? So this is licensing this transcription factor to display this sign. What's this going to bind to? We just learned. A nuclear something. What crawls through the nuclear pores? The nuclear import receptor. And that's all the thing it needs to get into the nucleus. That doesn't release it, but it gets into the nucleus because this thing is going to handshake with the nuclear import receptor. So it crawls through here. It gets in the nucleus. What causes it to be released from the nuclear import receptor? It's a switch. It's a G protein switch. It's called run, jump, ran. Okay? Ran GTP, license it to release the transcription factor with calcineurin still hiding the nuclear export domain. Okay? And so it's going to bind to all kinds of DNA and turn on and activate the T cell to do what its job is, which is to kill things that don't belong there. Okay, now that's a way oversimplification. So what happens is when this cell turns off, the signal will disappear, the calcium signal will go away, the calcium concentration lowers, and what happens is that causes the calcineurin to dissociate from the NF-AT, nuclear factor activated T cell. And so what's going to happen is that displays the nuclear export sequence. So now it's got a license to leave. And that's where we want to go if this is to be turned off. Okay? Simultaneously, you have to mask this little bump here. Otherwise, it's going to bind, it's going to stay in the nucleus. And so a kinase, which is unspecified by this cartoon, phosphorylates it and coats it with phosphate. So that masks the nuclear localization sequence. You've displayed the nuclear export signal because calcineurin has dissociated and revealed it. And so what's this going to bind to? To get out of the nucleus, let's use what we know. It's got to do what? What's going to read this? What's the code reading for this? It's an export, nuclear export receptor. Okay. But that's not good enough. The nuclear export receptor has to abound what before it can do that? RAN GTP, right? Because that's the rule. To get out, RAN GTP must be associated. So that heterotrimer then crawls back out, and then it encounters what on the fibril? RAN gap, which causes RAN to be shifted from, it loses the phosphate, so it goes from RAN GTP to RAN GDP. It releases the cargo and the export receptor back into the cytosol. Okay? And this returns to this state, which is off. Okay? All right, so you don't want to turn this off because it's a very important way of getting bad stuff that's attacking you, unless you're a transplant patient. Okay? So the problem is if you get a non-self transplant organ, what you have to do is suppress the immune system so it doesn't work. And what happens is, so the cyclosporin A, there's FFK, something, 205, 508, or whatever, are drugs. These are all anti-rejection drugs that suppress the immune system. And guess what they do? They inhibit this guy. So those cyclosporin A, which keeps a transplant patient from rejecting the transplanted organ is targeted to this protein here. And now you can see why. This cannot possibly be activated until calcineurin is available to mask the nuclear export signal and clip off the phosphates to reveal the nuclear import signal. So that's why they chose, this is a really brilliant choice, to design a pharmacological, inhibitors are always much more easier to design than activators. And so you just mess up calcineurin's function, and this can never make it into the nucleus to activate rejection of that tissue. Of course, it has side effects. Okay, any questions? So this is a very practical example of using this tool of gated transport into the nucleus and back out again. Okay. No questions? All right. Actually, the last thing I wanted to point out before we move on, and we won't return to this topic until we get to the cell cycle at the end of the course, actually. What happens is, in order to go through mitosis, what you have to do is you need to get rid of the nuclear envelope. And that's because microtubules, they're in the cytosol, right? And they have to project and bind to the chromatin, which is hidden by the nuclear envelope. So one of the key things in mitosis, it's actually prometaphase, is the nuclear envelope has to break down. How do you do that? Well, there's a really simple way. What you do is you phosphorylate the lamins. Remember, the lamins are the key to the structural integrity of the nuclear envelope. And so the trick is, all you have to do is phosphorylate the lamins. And what are lamins? They're cytoskeletal proteins that we will get to in about a month. Okay? And so if you phosphorylate them, they let go of the nuclear envelope. Simultaneously, they let go of the chromosomes. And so you get a twofer, right? You've gotten rid of the nuclear envelope. The spindle fibers can come in and attach to the kinetochores, and you can segregate the DNA, right? So that's a really efficient way to do it. But what's interesting is, you dissociate the nuclear pores and the nuclear envelope to little tubules that float around for a while. And then really all you have to do is, after the DNA has been segregated and you reform two new daughter cells, all you have to do is dephosphorylate the lamins. Okay? And this will self-assemble back into the nucleus. And one of the reasons it does that is the chromatin remains bound throughout this whole process to RAN-GAF. RAN-GAF remains associated. So when these tubules stick to the chromatin, they are by definition forming a nuclear envelope. And then these two fuse together, and finally you get this one big nucleus, and the transport of appropriate materials is going to occur because RAN-GAF is already trapped in the DNA. And RAN-GAP is only going to be located in the cytosol. It gets dispersed throughout the cytosol during mitosis. So so this system is designed to be broken up very quickly and be reassembled very quickly. And it's actually 30 minutes that takes. It's pretty cool. Okay, so that's all I want to say about getting rid of the nuclear envelope. Any questions about that? Yes?
You said it's 40 minutes or 4 minutes?
30. It's pretty quick. And if you're a tumor cell, you're going faster than that. Because that's all you want to do. And you don't even care if you get all the protein. One eyebrow, one leg is okay. As long as you can divide. All right, so that brings us to a different type of transport, which is, let me see how much time we have. About 15 minutes. Okay. A new type of transport. So we've done gated transport, and remember, it has nothing to do with gated ion channels or anything like that. It's very different. We're going to leave that. So now we're going to talk about a new direction that we have to go through, and that gets us into lecture 12. So we're going to talk about how does the ER involve itself in protein trafficking. And the ER is an amazing, actually a very amazing dynamic organelle that's way oversimplified. Usually you just show it as a couple of blebby sacs. It's really quite complex. So we're going to go from cytosol where a protein starts and somehow magically inserted into the ER. That's a complicated process called transmembrane transport. Okay, so this shows some of the dynamic nature. There's actually, so it's a series of networks of tubules and sacs, and there's all kinds of weird stuff going on in this compartment. I like this video, and this is the one I tested, so this really should work. I won't hold my breath though. See, let's make sure I'm. ER tubules? No, it's supposed to work. Yeah, it's something wrong with this. The way it's reading the thumb drive. Okay, so if you use the link, probably it would work on your computer. I'm not sure why this is not showing up. If not, you can go to the website that has all the full display and just take a look at it. And what happens, you can just see that all these tubules are forming and reforming based on which motor proteins bind to them. So what will happen is if you want to create a new connection between two tubules, a motor protein binds and just pulls out a little spindle of tubule and links it to a new place. And so you've got this really complex network that's very dynamic and changing all the time. It's not static in any sense of the word. So that was really the main message. Okay, yeah, this is a little bit better. Shows a little bit more of the complexities. Typically, there's two big divisions of the ER. That's the rough ER, where all the ribosomes are happening. And then there's the smooth ER, where you're blebbing off vesicles that are loaded with carbon. Okay? So those are the two big broad functions. Getting proteins into the ER so that they can go somewhere else. And where they're going to go is determined by what blebs off from the ER. Okay. So this is a cell that's, I think it may be a pancreatic acinar cell that's secreting pancreatic enzymes. I can't remember exactly what cell type it is, but it is obviously a protein secreting machine. So cells that are involved in lots of protein secretion, like your pancreas that secretes digestive enzymes, it's just loaded with rough ER. And the proportion of rough ER to smooth ER tells you what the function of that cell is. Okay? So if it's got a lot of rough ER, it's secreting the heck out of protein. And so you have lots of amplification of ER space. Okay? So let's go through some of the different functions of the ER. And co-translational entry of secretory proteins is one of the main functions of the rough ER. But that's not the only thing. Once you get the protein in there, and we'll talk about the exact mechanism probably next time, then things have to happen to it. The ER is really where the protein folds. Okay? The protein folding is important because in order to get into the ER, you have to unfold it. So proteins don't cross the ER folded. They have to be unfolded. So you thread it through this mechanism that we're going to talk about next time. But once it gets there, it has no function until it refolds, right? So fortunately, it's got several different proteins that are involved in folding it, okay? And so one of those proteins is called protein disulfide isomerase. And what that does is takes a protein like this and attaches these, what it does is it allows those disulfide bridges to form. What kind of environment must this be if those disulfides are forming? Oxidizing environment. So the ER lumen is an oxidizing environment that allows these sulfhydryl groups to join into a disulfide bridge. Okay. Now the problem is this may not be the lowest free energy state for that protein. So what happens is if it isn't, and say what if this disulfide needs to bind to this one, and this one really should bind to that one, and that would cause it to have a lower free energy state. What allows it to break those disulfide bridges to allow reformation? That's what protein disulfide isomerase does. It isomerizes this so it'll break these bonds and allow this thing to bind to that and this one to bind to that. And if it does, if it has the lowest free energy state, then it stops folding. And so the PDI, protein disulfide isomerase, is just allowing it to reform and refold into the lowest free energy state, which is what it's seeking. Okay? So that's a form of aiding a protein in folding. I don't think I have, yeah. The next kind is, there's a problem with proline residues. And we remember what that is. What does a proline do to the backbone of a protein? So if we have a sequence of proteins going like that, and then we run into a protein, what's it going to do? It's going to put a kink in it, right? Okay. Now, there's two ways to get a kink. Okay. This is cis. Both halves of the backbone go that way, or you can be going like this, and then it can be like that. So it comes in, it goes out a little different. It's called trans. So to go from a cis to a trans state is manipulated by this peptidyl prolyl isomerase. That's written down for you right there. So that's another thing. It's helping this thing kind of tweak the backbone. Sometimes it's better for it to be in a cis, and sometimes it's better for it to be in a trans situation. But it doesn't happen automatically, that protein cis, the conformational change. Okay? So that's a good thing to have. The third thing you have in your toolbook is this guy. It's called BIP, from a real, highly creative person, binding proteins called BIP. And what this is a member of the heat shock family, HSP family, okay? The heat shock family are proteins that are all ATPases, okay? Now what they do is that they bind to proteins that are exposing a hydrophobic patch to water, okay? And protects it from misfolding. And so what it will do is it will hide that hydrophobic patch, and then it will split ATP and release the protein to try to refold. If it folds properly, what's going to happen to those hydrophobic patches? Will they be exposed to the water or somewhere else? They will be hidden. And so when it no longer detects exposed hydrophobic patches, its job's over. So this chaperone, it's called molecular chaperone, they exist in the cytosol, but they also exist inside the ER lumen. The specific member of the HSP family, this is the family HSP70, that lives in the ER lumen is called BIP, okay? And there is a diagram in your book that's focusing on another HSP member. This one is when a protein is coming off the ribosome. Let's say it's going to be a cytoplasmic protein, like glycogen synthase or something, okay? That protein has got to fold properly, and what helps it do that are cytoplasmic heat shock proteins that are just basically called HS70 chaperones. Okay? So that you have them in the cytosol, but you also have this guy, which is located in the ER lumen. So when that protein comes unfolded into the ER, BIP can help refold it properly. So remember, BIP works on the ER lumen, but it's still a member of the same family that's helping proteins to fold. Okay, they're all ATPases. Okay. What other goodies do we have? Okay. One of the things that you need to understand is, what if you have a multimeric protein like the sodium pump? That sodium pump is going to go to the plasma membrane, so it's going to start its synthesis where? There's only one place, cytoplasm. And then it's going to go into the ER. Because it's got to go ER, Golgi, secretory vesicle, cell surface. So it's going to go into the endomembrane system. So it must use this transmembrane transport mechanism that we're going to talk about next time. And when it gets there, it's got 4 different subunits. So all the subunits come in independently. They've got to be reassembled somewhere. All of the multimeric assembly occurs in the ER. So it doesn't matter where you're going to do your function. If you're a membrane protein, your multimeric assembly happens in the ER. If it doesn't, you get rejected. You actually get thrown out and you're destroyed by the proteasome. So if you can't fold properly in the ER, you're destroyed. Okay? So this is why we say that the ER is the center of the quality control. Because if you don't fold properly and form the right kind of pump, you're discarded. And we have ways of detecting that we'll talk about next time. Okay, so you fold there, whether you're folded properly is determined, and there's a couple of diseases we already know about. One we've talked about, cystic fibrosis, is a misfolding problem. There's a mutation, one amino acid is deleted, I think it's 508, and that causes the whole protein to misfold so that it can never leave the ER. The protein's completely functional. It just doesn't ever make it to the plasma membrane, okay, which is really too bad. So the second type of disease that has that problem is at the basis of the pathology is inherited emphysema. And what happens is this protein is called alpha antitrypsin, okay? And what it does is it turns off a trypsin enzyme. Okay, so we need to step back and ask, what's the function of trypsin in the lung? Because if you have emphysema, that's got to be a problem with the lung. We talked about that mucus that's secreted outside the cell in the airway. We're using CFTR to actually deliver chloride, sodium, and water to rinse the mucus away. But some of it's so tightly bound, we need an enzyme to cut through the proteins, because mucus is just a glycoprotein. Okay? It's an extracellular glycoprotein. And what happens is your lung is secreting two things. It's secreting trypsin, which is a protease. It's chopping the mucus into little bits so it can be swept away and so you don't cough all the time. Okay? But the other thing that happens is if you let trypsin go by itself, it destroys the lung tissue. So you need this thing to stop the trypsin activity. And so it keeps the trypsin in balance. But if you have this inherited disease, this protein misfolds in the ER and it never makes it outside the cell to attack the trypsin that's chewing into your lung tissue. And so the lung tissue is damaged, not from smoking, which is the main cause of emphysema, but this is an inherited disease. So the take-home message of both of those examples is that protein folding is important, and the quality of folding is ensured in the ER before it ever goes any place. And that's, if you think about it, that's a very efficient place. Let's take care of it at the very beginning and then allow it to go somewhere else after we've said it could go. All right. Glycosylation, and we'll end the lecture with that. We're going to talk about that later, but glycosylation is a shared function between the ER and the Golgi, and I'll tell you how that works next time. Okay. But next time, of course, is the quiz number 2.
Question.
Yeah.
I was kind of confused on, you're talking about the ATPase, how it's a kinase and like it dephosphorylates. Like, how is that different from ABC? Like, how does that mechanism differ?
Why is the ABC transporter not a kinase?
Yeah.
It doesn't ever phosphorylate itself. It keeps the phosphate there and releases it sequentially, and then releases the ADP, but it never puts the phosphate onto a residue. That's why it's not going that distinction. Because it doesn't phosphorylate, it just cuts it.